Welcome back to the Sands Channel podcast.
Great to be here.
If you're enjoying these deep dives into the fascinating world of how our bodies work, please do take a quick moment to like this and subscribe.
Yeah, it really helps us bring you more of this content.
Definitely. So today we're embarking on, well, an incredible journey, really, to understand a pretty fundamental process, how our lungs develop those tiny air sacs that let us breathe.
Mm-hmm. And we're focusing on some surprising new research. It reveals this critical link between the energy powerhouses in our cells, the mitochondria, and making these essential lung cells.
That's right. This study really uncovers why certain metabolic processes, things happening at a really microscopic level, are just absolutely vital for building healthy lungs that work right from birth.
You might have some real aha moments, I think, as we unpack this.
Alrighty. Okay, let's dive in. So this research highlights the importance of alveolar cells. Why are these particular cells so critical for our lungs?
Okay, so think of your lungs like a tree, right? Branching out. At the very, very ends, the smallest branches, you have these tiny air sacs called alveoli. And that's where the magic happens, basically.
Yeah, breathing magic?
The breathing magic, yeah. The exchange of oxygen into your blood, carbon dioxide out. And this is mainly done by these super thin cells called alveolar epithelial type 1 cells. AT1 cells.
AT1. Okay, so they're built for that gas transfer, super thin.
Exactly. Their structure's just perfect for it.
So AT1s handle the oxygen and CO2 delivery. What about the other type, alveolar epithelial type 2, the AT2 cells? They have a different job, you said.
Totally different, yeah. AT2 cells kind of have dual function. First, they make this stuff called surfactant.
Surfactant, right. That stops the lungs collapsing.
Precisely. It's like a soapy film, reduces surface tension inside the alveoli, stops them sticking together when you breathe out.
Okay.
Second, and this is really key for the research we're talking about, AT2 cells act like the resident stem cells for the alveoli.
Stem cells, so they can make more lung cells.
Yep. They can divide and actually transform into new AT1 cells, which is obviously crucial for lung growth after birth, but also for repairing damage later on.
Ah, okay. A built-in repair kit. Makes sense. Now, the study mentions that how these cells, decide their fate, become AT1 or stay AT2, especially at the metabolic level, hasn't really been understood.
Right. That's been a bit of a black box.
And this is where the new research comes in, looking at mitochondria, the cell's power generator.
Exactly. The researchers zoomed right in on a specific part of the energy-making machinery inside the mitochondria. It's called the electron transport chain.
Okay.
Think of it like a production line, a series of steps making the cell's energy currency. And this study focused on the very first step, a protein complex called complex 1.
Complex 1. Okay, what does complex 1 actually do? Why focus on that one specifically?
Well, complex 1 has a couple of really crucial jobs. One is helping regenerate a molecule called NAD+.
NAD+, okay.
Think of NAD+ as like a helper molecule. It's essential for tons of metabolic reactions all over the cell. Complex 1 also pumps protons across a membrane inside the mitochondria.
Right, that's part of making energy, ATP.
Exactly. That proton pumping helps drive ATP synthesis. So complex 1 is involved in both energy production and this really vital NAD+ regeneration.
Okay, got it. Two key things. So how did the scientists figure out its specific role here in lung development?
They used a really clever trick with mice. They managed to genetically modify them so they could specifically delete a key piece of complex 1, this part called NDUFS2.
NDUFS2.
And they did this only in the lung epithelial cells and during pregnancy while the lungs were developing. So basically they knocked out complex 1 just in those developing lung cells.
Wow. And what happened to those mice? Sounds like it would have a pretty dramatic effect.
Oh, it did. It was fatal. The mice that lacked functional complex 1 in their lung cells, they died very soon after birth.
Oh, wow. What was wrong with the lungs?
Their lungs just hadn't developed properly. When the researchers looked closely, they found loads of these really abnormal cells. They seemed sort of stuck halfway between being an AT2 cell and becoming an AT1 cell.
Just the middle.
Yeah. They called them transitional cells.
Yeah.
It suggested the normal development pathway, the differentiation, was blocked somehow.
So no working complex 1 meant these crucial lung cells couldn't mature. Fatal. That really underscores how important complex 1 is. But how did they tease apart if it was the energy part or the NAD+ part that was the real problem?
This is where it gets really neat. The scientists introduced a different enzyme into these mice. It's a yeast enzyme called NDI1.
NDI1 from yeast. Okay. What does that do?
So NDI1 has this very specific talent. It can regenerate NAD+, just like complex 1 does. And this is the crucial bit, it does it without pumping protons.
Ooh. Ooh, clever. So it does the NAD plus job, but not the proton pumping energy job. That lets you separate the two functions.
Exactly. They could test if just restoring NAD plus levels was enough to fix the lung problem.
And what happened when they put NDI1 in those complex 1 deficient mice?
The results were pretty amazing, actually. Expressing NDI1 in the mice that were missing functional complex 1, it completely corrected the abnormal lung development.
Completely.
Yep. And the mice survived. They didn't die after birth.
Wow. So that strongly suggests it was the NAD plus regeneration, not the proton pumping for general energy, that was the critical function of complex 1 for these lung cells to develop properly.
Precisely. It points right at NAD plus regeneration being the essential task here.
That is a major discovery. It's not just about the overall power supply. It's this very specific metabolic task of complex 1 maintaining NAD plus.
Okay, the study also talks about something called the integrated stress response, or ISR. What's that? How does that fit in?
Right, the ISR. Think of the integrated stress response as like a cell's emergency alarm system.
A panic button. Yeah, kind of like a panic button. It gets switched on when cells experience different kinds of stress. Could be lack of nutrients, problems with proteins, or, relevant here, disruptions to their metabolism, like problems with mitochondria.
Okay, so how did they connect this ISR to the complex 1 problem?
They used a technique called single-cell RNA sequencing. It lets you look at which genes are switched on or off in thousands of individual cells all at once.
Very cool.
And they found that those abnormal transitional cells, the ones stuck in the middle in the mice lacking complex 1, they showed really high activity in genes related to the ISR. The stress response was definitely switched on in those cells.
So the faulty complex 1, or maybe the lack of NAD+, was triggering this cellular stress response. And was this ISR actually causing the developmental problems? Or just a side effect?
It really looks like it was contributing. The researchers tested this. They found that if they treated the mice with a drug that inhibits the ISR...
blocks the stress response.
Exactly. Or if they gave the mice a precursor molecule, something the body can use to make more NAD+.
Like topping up the NAD plus levels directly?
Right. Both of those things reduce the signs of the stress response in the lung cells. And, importantly, they partially rescued the mice from dying.
Partially rescued them, so it helped.
Significantly helped, yeah. It strongly suggested that this ongoing high-level activation of the ISR was a key reason the lungs weren't developing correctly. It was messing up the differentiation process.
Fascinating. So it's like a chain reaction. Faulty complex 1 low NAD plus triggers the ISR panic button. ISR interferes with normal lung cell development.
That seems to be the picture emerging, yes.
And didn't they also look at blocking a different part of the electron transport chain? Complex 2, I think?
They did, yeah. Another good control experiment. They looked at mice where they deleted a part of complex 2.
Okay. And complex 2 is also in that energy production line.
It is. It's involved in the electron transport chain.
Right.
But key difference, it doesn't directly regenerate NAD plus in the same way complex 1 does.
Okay. So what happened when they blocked complex 2?
Well, even though blocking complex 2 would also mess with energy production, they found it didn't cause the same super high level of ISR activation.
Interesting.
And it didn't cause the same really severe lung development problems and lethality that they saw when complex 1 was knocked out.
So that comparison really strengthens the argument that it's specifically the NAD plus regeneration by complex 1. That's the critical factor here, right? Not just general disruption of the energy pathway.
Exactly. It pins it down to that specific function of complex 1.
This is really painting a clear picture. Did the study look at when this was most important during development? Like at what stage?
Yes. They were careful about that. They timed the deletion of complex 1 to happen after the main airways, the bronchi, had already formed in the developing lungs.
Okay, so the main tubes were already there.
Right, and they didn't see major structural problems in the lungs before birth. The real issues, the abnormal alveolar development, started to become obvious after birth, specifically in those alveolar spaces where the AT1 and AT2 cells are doing their final maturation.
So complex 1's NAD plus role is particularly crucial for those later stages when the actual air sacs are forming and maturing.
Seems that way, yeah, for that final step of differentiation.
And you mentioned something earlier. There were some unexpected findings, too. It wasn't just cells dying off because they lacked energy.
That's right, yeah, that was quite surprising. They actually observed an increase in the total number of cells in the lungs of those complex 1 deficient mice.
More cells, not fewer.
More cells, yeah. And those cells seem to be dividing more, too, proliferating.
Huh. So it argues against a simple energy crisis, just killing the cells off.
Exactly. It suggested the main problem wasn't cell death due to lack of energy, but rather this block in differentiation. The cells were multiplying, maybe trying to compensate, but they couldn't mature into the correct AT1 or AT2 identities. They were getting stuck in that transitional in-between state.
Really stuck. And when they rescued the mice with that yeast enzyme, NDI1, that had quite a broad positive effect, didn't it?
Oh, absolutely. Putting NDI1 into the complex 1 deficient mice, it did more than just let them survive.
Yeah.
It pretty much normalized the levels of key metabolic molecules, like lactate, and importantly, it corrected that skewed NADH to NAD plus ratio.
Brought the NAD plus balance back.
Right. And like we said, it prevented death, it reversed the abnormal lung structure, the histology looked much better, and it even restored the lung's physical properties, like its elasticity or compliance, back to normal levels.
And that lasted.
Yeah, they checked long term, and the improvement held. It was a really effective rescue.
Okay, let's circle back to that single-cell RNA sequencing. You said it let them look at individual cells. What were the key details they got from that really deep dive?
So that single-cell view really allowed them to precisely identify and characterize that expanded population of transitional cells in the bad lungs.
The stuck ones.
The stuck ones, yeah. They could confirm these cells were expressing marker genes normally found in both AT2 and AT1 cells. Definitive proof they were in this arrested differentiation state. And interestingly, even the cells that looked mostly like AT1 cells in these deficient mice, they still showed some molecular signs of being transitional, not fully mature.
So the problem was quite widespread.
It seemed to be, yeah. And they also noticed changes in other lung cell types, like certain fibroblasts, suggesting maybe a broader disruption of how different cells coordinate during development.
And this all keeps pointing back to that integrated stress response, the ISR, as being kind of the central villain in this story.
It really does seem to be the key mechanism of the disruption. When they looked at the overall gene expression in the whole lung tissue, not just single cells, it confirmed increases in various stress pathways.
Right.
They saw upregulation of really well-known target genes of the ISR, things like AT4, ATF5, DDIT3, specifically in the lung epithelial cells lacking complex 1. And these ISR genes were most highly expressed in those transitional cells.
So the stuck cells were definitely stressed out, according to their gene expression.
Very much so. And when they compared the ISR signature in these transitional cells to similar transitional cells that you sometimes see during normal lung development or in models of lung injury, they found the ISR activation in the complex 1 deficient mice was significantly higher and seemed more sustained, less resolved.
So it wasn't just normal developmental stress, it was abnormally high and persistent.
Exactly. And they did this other cool analysis called RNA velocity, which kind of predicts the future direction of a cell based on its gene expression.
Okay.
And it suggested that these transitional cells in the complex 1 deficient mice were basically stalled. Their developmental trajectory had hit a wall, unlike the transitional cells in normal development, which progress onwards.
Stalled by the ISR, presumably. And they did experiments to really prove the ISR was causative, right? Not just correlated.
Yes, the crucial experiments, as we touched on when they gave the complex 1 deficient mice that drug ISRIB, which inhibits the ISR.
The ISR inhibitor, yeah.
It significantly extended their lifespan. They lived much longer. And it also improved the lung structure abnormalities.
So blocking the stress response helped fix the physical problem.
It did. And remember, giving the NAD plus precursor, NMN, also partially rescued them.
Right.
And both ISRIB and NMN reduced those ISR gene signatures. But here's a key detail. Only NMN, the NAD plus precursor, actually reduced the high NADH-NAD plus ratio.
So ISRIB blocked the consequences of the stress, while NMN addressed the likely cause of the stress, the low NAD plus.
That's exactly how it looks, yeah. It really reinforces that link.
Mm-hmm.
Low NAD plus due to complex 1 failure triggers ISR, and ISR activation blocks differentiation.
Okay. And did they check if this was all happening within the lung cells themselves? Like, was it an intrinsic problem, or did the cell's environment play a role?
Good question. They tested that using cell culture systems. Growing lung cells in organoids, which are like mini lungs in a dish, and also in flat 2D cultures. They found that differentiation defects were cell autonomous, meaning it was happening within the epithelial cells themselves. If they took normal AT2 cells in a dish and inhibited complex 1 chemically, it prevented them from properly differentiating into AT1-like cells, just like in the mice. And they suspected this was because inhibiting complex 1 directly triggered the ISR within those isolated cells.
Makes sense. So the problem travels with the cell.
It seems so. And they even started to pinpoint a potential molecular pathway inside the cell involving proteins called OMA1, DALE1, and HRI. That might be the specific sensors linking complex 1 dysfunction to switching on the ISR in these lung cells.
Wow, getting down to the molecular triggers. And just to wrap that comparison up, the complex 2 inhibition findings really drive home the NAD plus point, don't they?
Absolutely. It's worth restating because it's such a key control. Inhibiting complex 2, which is part of the energy chain, but doesn't do that direct NAD plus regeneration, it just didn't cause the same catastrophe. No massive ISR activation. No fatal lung phenotype. It really isolates the NAD plus regeneration by complex 1 as being uniquely critical here for preventing this harmful sustained stress response and allowing normal alveolar development.
So if we boil it all down, the big takeaway is that complex 1 isn't just about cranking out ATP energy. Its specific job of regenerating NAD plus is absolutely essential for lung air sacs to develop properly. It does this by preventing this runaway cellular stress response, the ISR, from getting stuck in the on position.
That sums it up perfectly. It's a really novel insight into how mitochondria, beyond just being power plants, play a critical role in determining cell fate and telling these lung cells what to become during development. It links metabolism directly to cell differentiation via this stress response pathway.
That's really fascinating stuff. So what does this all mean, maybe bigger picture? This research gives us this really fundamental understanding of how lungs develop, highlighting this delicate metabolic balance.
Yeah, and it definitely raises questions, Could targeting the ISR, or maybe boosting NAD plus levels, be potential therapeutic strategies for lung developmental disorders, or maybe even for helping lungs repair after injury in adults?
Interesting possibilities. What really stands out to you from this deep dive?
For me, it's just that unexpected role of NAD plus regeneration being the critical factor from complex 1 in this context. It highlights how specific metabolic functions, not just overall energy, can direct major developmental processes, and also how sensitive development is to sustained stress responses.
Yeah, it underscores that incredible complexity inside our cells, how seemingly small mitochondrial jobs can have such profound effects on how our organs form. It definitely encourages you to think more about mitochondrial metabolism and cell signaling.
Absolutely. There's still so much to learn there.
Well, this has been incredibly insightful. Please don't forget to like the video and subscribe if you want more content like this.